In this video, I'm going to briefly talk about some of the things going on with the National Institutes of Health funding gain-of-function research in China at the Wuhan Institute of Virology. And so a lot of this is based on findings that are published on the Intercept, and I'll have links to all the articles below, by Sharon Lerner and Mara. I can't pronounce that last name. So I think it's mostly Sharon Lerner. Yeah. So this one is Sharon Lerner and two other people, and this one is also Sharon Lerner. But anyway, there has been a lot going on with this. As you can see, they have more than 900 pages of materials, and so I'm not going to sort of go over all of that stuff, especially since if you are interested in all of that stuff, then I would recommend checking out these Intercept articles. What I am going to do is go over a grant proposal that is talking about the kind of research that this institute called the EcoHealth Alliance. So the EcoHealth Alliance has been getting funded for by the American taxpayers and doing in collaboration with Chinese institutions, including the Wuhan Institute of Virology, and there was one other one. So yeah, the Wuhan University Center for Animal Experiments. So that's another one that has come up in this discussion. So I'm going to be looking at this grant proposal, which was also obtained by the Intercept. And so one thing to note is that this particular grant proposal was not actually funded. So this is a grant proposal to DARPA, and this one was not actually funded, but this is from EcoHealth Alliance, and this is the kind of experiments that is being talked about in all of the other things. And so I'm going to go through a little bit of the science from this, just so maybe you can understand what the experiments actually are. And so I've highlighted some stuff throughout this that I'm going to be kind of skipping through to the relevant things. And so, well, I guess I should say that this is a grant proposal, and those take on sort of a standard way of presenting information and, well, asking for money. And so this one is broken up. So there's this executive summary, which is, I guess, sort of the overall sort of summary of what they are trying to do. And so it's somewhat like an abstract on a scientific paper. And then this one, then they go to this next section, which is the goals and impact, which is essentially trying to say, hey, this is what we're going to get out of this research if you fund us. This is why you should research us. And then there are these technical areas, which in some grant proposals they're called aims or something like that. This one calls them technical areas, and this has two technical areas. And the first one is sort of the relevant one that I'll be going over the most, and that's the one where they're actually obtaining viruses from bats at in different parts of China and doing experiments with them. So one of the things they're trying to do in the first technical area, or the first aim. So they're essentially trying to do two things. The first one is fairly, I guess, innocuous as far as whether it's dangerous to people, and in fact, it's probably one of the kinds of things that people would really like scientists to do, and that's just sort of tracking the coronaviruses that bats are carrying and finding out where those coronaviruses are and how far they're spreading and if they're sort of being transmitted to other bats in other parts of the country. And so that is sort of the first part of the first technical area. The second part is the part that is, and I'll be talking about this in a little bit more detail here in a little bit, is the part that is sort of concern, and that's where they're actually taking the spike proteins from the coronaviruses they find and inserting them into a different coronavirus backbone in order to test its sort of pathogenicity against humanized mice. And so the humanized mice being mice that express the human ACE2 receptor rather than the mouse ACE2 receptor. And so that is the part that is sort of concern to people. So then technical area 2 is, once again, this is not any of the gain of function type research, and it's actually looking at ways of sort of, I guess, boosting or activating the bats' immune system so that the bats themselves could actually fight the coronaviruses and reduce the risk that they pose to humans, reduce the risk of the virus spilling over to humans. So now let's sort of go through this in a little bit more detail. So I highlighted this up here. We will sequence the SARS coronaviruses, spike proteins, and reverse engineer them to conduct binding assays and insert them into bat SARS related coronaviruses. So that's the WIV1, which stands for Wuhan Institute of Virology, because that's where this one was discovered, or this SHC014 backbone. And they, in parentheses, these use bat SARS-related coronavirus backbones, not SARS-CoV, so the SARS-CoV-1 from back in like 2003, and are exempt from dual use and gain of function concerns. So they are saying that doing this experiment is not gain of function. So maybe from, some technical point of view, it could be considered not gain of function, but there are probably a lot of people who would interpret this as gain of function. We'll go into that in more detail here in a little bit. To infect humanized mice and assess capacity to cause SARS-like disease. And so that right there, so if we define gain of function as essentially increasing either the pathogenicity or the transmissibility of a virus, the pathogenicity being, how dangerous it is, how much damage it actually causes. They are looking to see if doing these experiments actually increases the pathogenicity of the virus, which seems like it would fit the definition of gain of function. But like I said, they say here that these use the bat SARS related CoV, which is this WIV1 and SHC014 backbones, and not the SARS-CoV-1, I guess you could say. I think they sometimes call it SARS-CoV urbani or something like that for that first SARS outbreak back in 2003. And so they're saying that these are exempt from the gain of function concerns, which means that there must be some, technical definition in the National Institutes of Health that say that only if they're inserting it into the SARS-CoV-1 that it would be gain of function. But, that's sort of the, I guess, political definition. But, if we're actually trying to come up with a definition, the concern is this actually gain of function? And so I highlighted this just because it does show that Dr. Shi at the Wuhan Institute of Virology will conduct viral testing on collected samples and binding assays on some humanized mouse work. So this grant, which once again, this grant was not actually funded. Other grants have been funded doing pretty much these same types of experiments, and that those are, in that large, 900 pages of documents that the Intercept received. But anyway, so in this one, they wanted to use the Wuhan Institute of Virology to do some of their experiments. And so that's what that is there. So now we can go to the technical area 1 here. And so this whole paragraph, and I'll kind of read through this and comment on it. So to characterize spillover risk of SARS-related CoV quasi-species, and they'll use this QS abbreviation here for quasi-species. And so that's essentially a possible future version of a virus that could potentially spill over. And so they want to look at the spillover risk of these possible future SARS-related coronaviruses. The Wuhan Institute of Virology team will test bat fecal, oral, and blood samples for SARS-related CoVs by PCR, which is PCR is just a technique for amplifying segments of DNA and sort of seeing what DNA is actually there. We will collect viral load data from fresh fecal pellets. SARS-CoV spike proteins will be sequenced. Viral recombination events identified. So they're actually looking for when recombination actually happens in the wild, which is something that does happen, especially with viruses and bacteria. A lot of recombination events can occur, and so that is when parts of a genome from one virus can actually combine with parts of a genome from another virus, and essentially nature doing its own gain of function sort of things. And so that's the thing that people are worried about, is that nature will do sort of a quote-unquote gain of function and create a virus that can then spill over into humans. And so that is sort of the logic behind doing gain of function research, is to find these recombination events that could occur in nature in the future and then anticipate them. So that's the logic behind gain of function research, is to anticipate sort of quote unquote gain of function that nature would just do naturally. So anyways, SARS-related coronavirus spike proteins will be sequenced, viral recombination events identified, and isolates used to identify strains that can replicate in human cells. So looking for, that's just looking for isolates for viruses that can actually infect humans. The University of North Carolina team will reverse engineer spike proteins of a large sample of high and low risk viruses for further characterization. This will effectively freeze the QS, so that's that quasi-species. We analyze at t equals 0 these QS sub 0 strain viral spike glycoproteins. So it's called a glycoprotein because it's a protein that has sugars sort of added onto it. And sugars are often a way for different receptors to identify a protein. In fact, if you probably heard of things like the blood types, like the A, B, and AB and O, those are talking about different sugars on the receptors on red blood cells. So different sugar combinations. So that is how those proteins are recognized. But anyway, these QS sub 0 strain viral spike glycoproteins will be synthesized, and those binding to human cell receptor ACE2 will be inserted into the SARS-related coronavirus backbone, so into that WIV1 backbone, for instance. So that is essentially, they have the viral genome here. So this is the viral genome. They take the spike protein out of it, and then this right here is the SARS, or the WIV1. This would be the WIV1 IV1 backbone. So this is the spike protein right here, or the spike protein gene in this CoV genome here, genome. And so then they'll take this spike protein and actually insert it into this WIV1 backbone. And so the logic behind this is actually to sort of isolate the spike protein and see what difference the spike protein itself is making, because they know the infectivity of WIV1. So WIV1, which has its own spike protein here. So if this is the WIV1 here, it has its own spike protein, and they know what the pathogenicity of this is, the pathogenicity of this WIV1 is. So if they remove this spike protein and then insert a spike protein from another newly discovered coronavirus, so we could call this the QS here, then they can look at how inserting this other spike protein into the WIV1 changes, so how that changes the pathogenicity by putting that into humanized mice, mice that express ACE2 in their lungs, and see how much that changes the pathogenicity of the WIV1. So that's the logic is that is looking at just how much this new spike protein is actually changing the pathogenicity of the WIV1, and so that will tell them, that will give them information about that spike protein there. So the QS sub-zero strain viral spike glycoproteins will be synthesized. Okay, so binding to human cell receptor ACE2 will be inserted into the SARS-CoV backbones, so the WIV1 backbones. So they're saying here again that is non-gain of function. They are saying that by the definition they are going by, that is non-gain of function. Then, like I said, inoculated in human mice to assess capacity to cause SARS-like disease, so to test the pathogenicity, as well as the efficacy of monoclonal therapies and other different types of therapies. And so this is technical area 2, which didn't really have anything of concern. So then they go through the technical plan, which is what they, sort of more detailed what they plan to do for each of these. So once again, using this WIV1, WIV16, and SHC014, that unlike other SARS-related coronaviruses, do not contain two deletions in the receptor binding domain of the spike protein. So the part of the spike protein that actually binds to the human ACE2 receptor. They have far higher sequence identity to SARS-CoV-1. So they are closely related to the SARS-CoV-1, which was the 2003 outbreak, use human ACE2 receptor for cell entry. So they're saying that these SARS-related coronaviruses do actually use the human ACE2 receptor for cell entry, as the SARS-CoV-1 does, and replicate efficiently in various animal and human cells, including primary human lung airway cells, similar to the SARS-CoV-1. So chimeras, this is the recombinants, this is the sort of gain of function part of it. With these SARS-related coronavirus spike genes inserted into a SARS coronavirus backbone and synthetically reconstructed full-length of these SARS-related coronaviruses cause SARS-like illness in humanized mice. So in this instance, they're actually taking the spike proteins from the SARS-related coronaviruses. Well, I don't think they are doing it. They are citing two papers that have done this. So they're actually taking the spike proteins from the WIV1, for instance, and putting it into the SARS-CoV-1 backbone. Uh, and then they found that this caused illness in mice with clinical signs that are not reduced by SARS-CoV monoclonal antibody therapy or vaccination. So they are putting the WIV1, for instance, backbone into the original SARS-CoV-1 or SARS-CoV Urbani, I've seen it called. And they find that that also causes illness. And once again, this, I think, is not something they are doing. This is something they are citing here in this reference 11 and 12. But that caused SARS-CoV-like symptoms in mice expressing human ACE2. All right, so this is all that sort of stuff I was talking about, where it's just sort of tracking where the coronaviruses are, and you can see they have this map here of China, and they want to be able to track where these coronaviruses are. And then this part here, this prototype app for the warfighter, they have to sort of justify why DARPA would be funding this by sort of saying, hey, this will be helpful for, soldiers and fighting wars and stuff. But anyway, we'll go here. So the SARS-related CoV QS detection, sequence, and recovery. We will screen samples for SARS-related coronavirus nucleic acid using our pan-CoV consensus one-step hemi-nested RT-PCR assay. I thought about going into detail about what this one-step hemi-tested RT-PCR. I'll just sort of give a quick overview. So one step meaning that they are doing it all in the same PCR. They don't have to open the lid of their PCR machine and do anything. It's all in one step. Hemi-nested is interesting because they're essentially using two different sets of primers for their PCR. And nested meaning that one set is sort of outside and then the next set is sort of inside the sequence of that. Then RT-PCR in this instance is reverse transcriptase PCR. Sometimes RT-PCR means real-time PCR. In this instance, it's reverse transcriptase because they're taking the viral genome, which is RNA, and turning it into DNA, which is what the reverse transcriptase does. But anyway, they'll be looking, they will screen samples of the SARS-related coronavirus nucleic acid using this technique, targeting a fragment of the RNA-dependent RNA polymerase of all known alpha and beta coronaviruses in specific assays for known SARS-related coronaviruses. Essentially, what this is doing is they're looking at this this RNA polymerase because that's going to be shared among all these different coronaviruses, so they can detect the presence of coronaviruses using this technique. But then I have down here in red, so synthesis of chimeric novel SARS-related coronavirus quasi-species. We will commercially synthesize the SARS-related coronavirus spike glycoprotein genes designed for insertion into these two backbones here. So these SARS-related coronavirus backbones, molecular clone backbones. So 88%, 97% spike protein identity epidemics. Yeah, so SARS or Bonnie here, that's the one I've been talking about. It's the original SARS-CoV-1. These are BSL-3 not select agents for subject of the P3C0, which I forget what that actually stands for, but it's essentially uh, the thing that's overseeing or defining what gain of function is. And so once again, they're saying that they use these bat SARS-related coronavirus backbones, which are exempt from this definition of gain of function, and are pathogenic to the human ACE2 transgenic mice. So they are making these chimeric versions of coronaviruses that are pathogenic to human ACE2 transgenic mice, so mice that express the human ACE2 receptor, but they're saying that these are exempt from this definition of gain of function. And so, once again, it's one of those things where it's like, is the legal definition the definition we ought to be using for gain of function? And so, yeah, I mean, you can judge that, I guess. All right, so then here, mutations in the receptor binding domain, the host protease proteolytic processing of the S-glycoprotein, so that's that furin site that is found in the SARS-CoV-2, regulate the SARS-related coronavirus cell entry and cross-species infectivity. Mismatches in the SARS receptor binding domain ACE2 molecules or S-proteolytic processing will prevent entry of SARS coronavirus, and their quasi-species with these mismatches will be deprioritized. In other words, they are only interested in the coronaviruses that can infect human cells. And so ones that can't, they're going to deprioritize, which means they're not going to worry about studying those, which, you know, it makes sense in light of the logic they're using here. They're interested in coronaviruses that can infect humans. So they're not interested in studying coronaviruses that don't infect humans. So single amino acid variations could dramatically alter these phenotypes, the phenotype being here that it can infect human cells. And we will evaluate the impact of low abundant, high consequence microvariation in the receptor binding domain. So low abundant, there aren't very many of them. High consequence meaning that they are particularly infective, using RNA-Seq, which is a technique of actually finding RNA in the cells. So it's often used in studies that want to look at what's called the transcriptome. So the genome is the DNA in the cell, and so that's looking at all the genes in the cell. The transcriptome is all the genes that are being expressed and to what extent they're being expressed. And so RNA-Seq is a technique that can actually look at sort of how much each gene is being expressed by looking at the abundance of the mRNAs for those genes in the cell. But in this instance, they're using it to look at the amount of viral RNA in the cells. So that's how they can identify the low abundant quasi-species variants, by looking at how much of the RNA with a particular mutation is there and finding the ones that are low abundance, encoding mutations relevant to the ACE2 binding. We will conduct in vitro pseudovirus binding assays using established techniques and live virus binding assays. And so I highlighted in red here at the Wuhan Institute of Virology to prevent delays and unnecessary dissemination of viral cultures. Essentially meaning that they want to do it there in China where they have the Wuhan Institute of Virology, so they don't have to be sending these samples, back to the United States in order to do this testing. But The reason I highlighted it here in red is because it does show that they are working in collaboration with the Wuhan Institute of Virology. And then so here, so this is kind of a long section, but it's sort of the uh, much more in technical detail what they're actually going to be doing here. So we will synthesize quasi-species with novel combinations of mutations. So they're going to be using novel combinations of mutations, so new mutations, to determine the effects of specific genetic traits and the jump potential of future and unknown recombinants. So the first part here, which I have in this sort of darker yellow, is the receptor binding domain deletions. So um, I won't go through that in detail here, but they're going to be looking at essentially changes in the part of the spike protein that actually interacts with the human ACE2 receptor, because that interaction is going to change the pathogenicity of the virus towards humans. Then, of course, there is the proteolytic cleavage site, which is that, and SARS-CoV-2 is that furin cleavage site. And so, that there are other types of proteolytic cleavage sites that other coronaviruses use. What was interesting to people about the SARS-CoV-2 is that it used this furin cleavage site, which wasn't found in other closely related coronaviruses to the SARS-CoV-2, which is one of the things that makes people think that that the SARS-CoV-2 is actually leaked from a lab rather than through a natural recombination event. So after receptor binding, a variety of cell surface or endosomal proteases cleave SARS-CoV spike glycoprotein, causing massive change in the spike protein structure. And I've talked about this in previous videos about I've looked at the actual structure of the spike protein and the sorts of changes that actually come about from this cleavage, and how that actually allows entry of the viral particle in the viral genome into the human cell. So I won't go over that here. Activating the fusion-mediated entry. We will analyze all SARS-related CoV spike gene sequences for appropriately conserved proteolytic cleavage sites in the S2, which is sort of the part of the spike protein. There's S1 and S2, so after it's cleaved, it's in two parts, and one of them they call S1 and one they call S2. And for the presence of potential furin cleavage sites. So they're looking at wild coronaviruses for potential furin cleavage sites, because that furin cleavage site would allow entry into human cells. And so they're worried about furin cleavage sites coming about in the wild, because that would be a condition, a sort of function that these viruses could gain in the wild that would allow them to become more pathogenic and more virulent to humans. So SARS-related CoV spike with mismatches in proteolytic cleavage sites can be activated by exogenous trypsin. And so they're just saying that that trypsin can cleave these as well. Trypsin is a digestive enzyme actually that breaks down proteins, and so it's not surprising that trypsin would be able to cleave this. Where clear mismatches occur, we will introduce appropriate human-specific cleavage sites and evaluate growth potential in viro cells and cultures. So they're saying here they will introduce appropriate human-specific cleavage sites, which is essentially what people are worried about with the SARS-CoV-2 having this furin cleavage site, that humans have introduced the the SARS or the fear and cleavage site into this coronavirus, and that's what made it more infective and more pathogenic and more virulent to humans. And so uh, this they're actually saying that they are going to introduce those human specific cleavage sites and evaluate the growth potential in these cell cultures. In SARS-related CoV, we will ablate several of these sites based on pseudotype particle studies and evaluate the impact. So essentially, they're going to also do loss of function studies here, is what they're saying. We'll also review deep sequence data. Then there's also this part, which I have in green, the end-linked glycosylation. So glycosylation is that adding of sugars to these proteins. So some glycosylation events regulate SARS-CoV particle binding. Alternative receptors for SARS-CoV entry into macrophages or monocytes, which are both types of white blood cells. So mutations that introduce new end-linked glycosylation sites, so places where these sugars can bind, may have been involved in the emergence of human SARS coronavirus from civet and raccoon dogs. And so they're saying that essentially it's a possible pathway for the original SARS from 2013 going from bats to humans was through civets or raccoon dogs, and it was in these civets and raccoon dogs that actually caused a change in these glycosylation sites, which so essentially it spilled over from a bat into a civet or raccoon dog, and then in the civet or raccoon dog, that's where these new glycosylation sites came about, which allowed spillover into humans. While the sites are absent from civet and raccoon dog strains, uh, well, I'm not going to go through this. It's not really of what we're interested in here. And so this, it's hard to see on here, but this is sort of the flow chart of their work here. And so it's kind of hard to see here, but this blue one right here, presence of proteolytic and furin cleavage sites. So blue being a predictive trait, and then you can see that from there they will look at cell entry. And anyway, so you can look at this paper yourself if you want to look at sort of the flow chart of what their experiments are actually going to be. But I just wanted to point out that the presence of proteolytic and furin cleavage sites is here. Then looking at cell entry and intracellular growth and pathogenesis in new hosts. And so, yeah, that is the flow chart for that. Then technical area 2, which as I said didn't really have anything that was too worrying as far as gain of function research. It was looking at, as I said, essentially looking at ways of increasing, well, it's not genetically manipulating bats. It's sort of like giving bats, they're looking at ways of giving bats a vaccination against coronaviruses, which would reduce spillover potential into humans. Then here they're kind of saying that this could also be the sort of science they're doing here could also be used against things like Ebola and other diseases. And so that's kind of one of the things they're using to sort of appeal to DARPA and saying that, well, this could help with with other diseases and other areas that we are interested in sort of militarily. So dynamic circulation modeling to optimize deployment strategy. So like I said, they're appealing to DARPA, which is a military science program. So this is the management plan. These are all the scientists who are sort of involved here, and so we see a lot of these usual suspects. So Peter Dajak, I don't know how to pronounce his name. It looks maybe Polish. But he is one of those scientists. He's in charge. He's, I think, the president. I don't know if he's the president. He's a big name in the EcoHealth Alliance, and he's the principal investigator uh, involved in a lot of these EcoHealth Alliance science. So you can see down here, he's also in the technical approach one here. Also with Zhengli Shi at the Wuhan Institute of Virology. We also see Dr. Ralph Barrick, who's at the University of North Carolina. He is also a usual suspect in a lot of these sort of iffy gain of function type experiments and so forth. So down here, once again, I've just sort of highlighted. So we have Peter Daszak. We have Dr. Zhengli Shi, who's at the Wuhan Institute of Virology. And it looks like the University of North Carolina is, yeah. So yeah, these are biographies of some of the scientists involved here. We got the EcoHealth Alliance. We got the Wuhan Institute of Virology here. So this is just another sort of little summary here of all the different tasks. So once again, I've highlighted the ones that are sort of interest here. So this PITA-01 Task 3, screen and characterize and isolate SARS-related coronavirus quasi-species from bat samples at the Wuhan Institute of Virology, PCR screening. So subtask 3.3 is the one that to develop and recover recombinant viruses with spike proteins and novel coronaviruses. So they're saying that that is going to be done at the Duke NUS. Then, of course, down here we have the experimental assays with those. So that's all just kind of what we've already gone over. I don't know if I had anything else to talk about here. Oh, yeah, so this is the risk mitigation plan, which interestingly does not really have anything about the potential gain of function experiments. It's all about their animal use safety and field safety and things like that. They have the ethical, legal, societal implications, which once again doesn't really talk about any of the potential gain of function things. And then so there's a bibliography here. But anyway, that is the proposal for this diffuse project here. And so, like I said, this will be linked to in the description down below if you want to go through it in more detail yourself. And like I said, there are a bunch of other things that have been just found out. So this is the thing here, which is from the Intercept article. You can see this is the notice of award. So these are things that the National Institute of Allergy and Infectious Disease, which is part of the National Institute of Health, actually sent to the well, see, you can see here Peter Daszak and to the EcoHealth Alliance. So these are things they sent back to the EcoHealth Alliance, essentially saying, we are awarding you this money. And so these are for different proposals than the DARPA one I was just looking at, but they are doing pretty much the same experiments that I was just talking about. And so, I mean, this is all a bunch of, technical jargon and legalese, and it's, 528 pages of it. So that's why I didn't really feel like going through all of this. But anyway, that will be linked to in the description down below if you feel like going through it. There was one thing when I was looking through it um, that talked about sort of safety measures. Well, once again, you can see here the Wuhan Institute of Virology. But anyway, I mean, it did stipulate at one part, I can't find where that is, that essentially, if you find out that, essentially that you're doing gain of function research, then you have to stop and, tell us about it. And uh, I believe the Intercept articles have said that, they did not do that and sort of persisted in doing these potentially dangerous experiments. But anyway, I've been kind of droning on long enough. I hope you found this video helpful, at least in understanding the science part of it. I don't really feel like going into the sort of politics of it and trying to discuss should we define this as gain of function and all that. But anyway, I hope you found this useful, and I will see you in another video.